Oke, Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Welcome to our International Guest Lecture Series organized by the Faculty of Pharmacy, Universitas Indonesia. We are honored today to have three distinguished speakers with us from Nara Institute of Science and Technology, Japan. But before we begin, may I kindly request for everyone to set their mobile phones to silent mode to avoid any interruptions during the event. Thank you. And allow me to introduce myself. I am Donna Mareta Aryastanti, and I will be your moderator for today's event. And I am pleased to introduce you to our three distinguished speakers that will present their research. First is Professor Yasumasa Beso, um, who will present his research with the title Valproic acid induces axial skeleton anomaly via perturbations of the somite segmentation clock. Welcome, Professor, and it's an honor for us to have you here with us. And then the second speaker will be Professor Naoki Aratani, um, who will present material science at NAIST from synthetic nanocarbons to degradable polymers. And then last but not least, of course, Professor Hisaji Maki, who will introduce us about NICE and all the postgraduate programs in NICE. Okay, without further ado, we will hear the explanation about NICE and their postgraduate program from Professor Hisaji Maki. Please welcome, ladies and gentlemen, Professor.
Thank you. My name is Maki, and I'm very glad to be here. And yeah, so many students gather here. And let's see, I needed to share. It's already shared. Yes. And let's see. Oh, no. What I should do that. Maybe not. It's this one. Much better. It's okay. So my name is Hisaji Maki. I'm a professor emeritus of Division of Biological Science, Nara Institute of Science and Technology, called short in Nara, NAIST. And I've been working on the international exchange and recruitment of the international students from our partner universities for many years. And I like to introduce you the NAIST and the Division of Biological Science, NAIST. Okay, so first thing is, where is NAIST? So this is a Japan island. The Tokyo is here. So from Tokyo, 500 kilometer west, the Nara located here. And it's very close to Osaka, big city, and Kyoto, very old city, beautiful touristic city. Like 30 minutes by subway to Osaka, and 1 hour by train to Kyoto, very close. And Nara itself is very peaceful, quiet, nice city, famous for the big Buddha and deer park, right? So our institute locates to the next city of Nara, okay? And please remember, NAIST is the youngest national university in Japan. The last national university settled in 1999. And also the smallest national university in size in Japan. I will show you some evidence for that. So this is a photo of our campus in the evening, and established in the 1991 as the 84th National University. It's the last one, okay? And the unique feature is this university is only for graduate students. So NAIST doesn't have undergraduate, okay? So only graduate school, graduate students. And we have three areas focused on: biological science and information science, IT technology, and material science, and chemistry, physics, such kind of advanced research studies. And you can see very compact campus, okay? So maybe, let's say 100th smaller than UI campus, or maybe more than that, okay? It's very small. And let's see, the number of students, we have master students 760 and doctoral students about 400. So total number is like 1,200 students, are all postgraduates, with the faculty 200. So number of faculties are relatively high, and 1,000 one professor and the students. There's such kind of structures, and very small, the young and very small. And then we are the most highly evaluated national university in Japan. So every 6 years, Japanese government examine and evaluate all national university, 84 national universities in education and research. The NAIST was evaluated as excellent. Okay, another university evaluated excellent was Tokyo University of the Art. It's not the University of Tokyo, big university. This university is for music and painting arts or other arts, right? So for the education, that Nara Institute of Science and Technology provides a very, very high quality that is evidence, and the research that is also evaluated as excellent. Okay, this case research, other universities like University of Tokyo or Kyoto University, many other, but NICE is only one. The both evaluated in both education and research are top. Okay, so that's one evidence for that. And another evidence is the Thomson Reuters essential science indicators and Japanese newspaper companies, the university ranking in Japan, the overall ranked first. Okay, NICE is ranked first, especially the plant and animal science is ranked first. Okay, second is Tokyo University, University of Tokyo, and third one is Kyoto University. We are much higher. Okay, and chemistry ranked third. Okay, unfortunately, the chemistry is not the top, but it's very high. And the molecular biology and genetics ranked third, and microbiology ranked third. This case, the Riken is the top. Riken is the National Institute of Science. Okay, so Riken is the top, and the Tokyo University is the second, and we are the third. So the let's say youngest, smallest, and the very strongest university in Japan. Okay, remember it. So I will explain Division of Biological Science. We have 24 laboratories. Okay, in quite variety of the research areas. Today, with the 100 faculty members and 250 master students and 100 PhD students. And in this PhD student, 50 are international students and 12 Indonesian students. Okay, so many. So you can see many international students from ASEAN countries, Malaysian or Thailand, Indonesian, and Vietnamese. Okay, and let's see. As I told you, that NAIST biology is the top graduate schools in biology in Japan. And we made excellent research and activities and achievements. And we have the excellent teachers and education program. So one of the former faculty member is the Professor Shinya Yamanaka, who invented the technology to create the stem cells from normal cells, called the iPS technology. He didn't do that. His students, NAIST students, developed the iPS technology. Okay, but Professor Yamanaka wanted to apply the iPS technology to the human medicines or medical science, okay, to let's say rescue that the human health or something. So they moved to Kyoto, but actually he spent 7 years at NICE, and we have many faculty members like Professor Shinya Yamanaka. Okay, so let's say our teachers are very excellent, and we are actually rich in the research fundings. This graph shows how much money got by one researcher per year. So the number one is Tohoku University, because they have big facility for linear accelerator or something like that. It's a big university. So they have a big research facilities. It's a huge amount of money takes. And Tokyo University and Osaka University also has a big facility. So it doesn't mean that this amount reflects the actual research funding. Okay, but NICE doesn't have such a big facilities. Okay, so NICE is the richest university in terms of the research funding, and we have many cutting-edge facilities, very fancy microscopes and electron microscopes and many other analytical machines. And let's say I'd like to explain what kind of research are doing in divisions. We have 3 areas. One is the plant biology, plant science. Okay, I think plant biology is the most strong in Japan, and it's very competitive to other famous institute in the world. Okay, so plant scientists, everybody knows NICE, plant biology. We have 9 laboratories. Oh, sorry. Oh, something happened. I don't know. And the second area is medical biology, biology related to medical things, okay? Medical things, medical biology, 7 laboratories. And the third one is systems biology, we call, which includes the microbiology and structural biology and bioinformatics, such kind of things, okay? And I picked up, these are 9 laboratories of plant biology. You can access this information by accessing our website. Okay, we have in English version, so you can tell more precisely or more deeply what kind of science they are doing. And I picked up two laboratories. One is a plant growth regulation by Professor Umeda. This laboratory studies the genome maintenance of plants and DNA polyploidizations. Plant sometimes double their genome, okay, or sometimes triplicates or tetraplicates to expand the size of cell or size of plant. So he is studying the mechanisms, how the plant control this such kind of uh, doubling or uh, more the genome, and it relates to the mechanisms to maintain the genome. Okay, and this technology is very important to produce more crops, or let's say the makes the bigger plants. Okay, so use his technology. He made a 10 times bigger strawberry. Okay, so usual strawberry is like this, and the bigger strawberry is like this, very big. Okay, so he is an expert of this kind of science. And next one, the Professor Ito studying the how plant controls the flowering. Flowering is the key process to make uh, crops or seed. Okay, so the flowering the should be studied to control how when harvest the vegetables or harvest the crops, right? So it's a most molecular level things. The what kind of genes are involved, or what kind of let's say signal actions or transcriptional controls are involved in this process. So yeah, this is very key key study to improve the production of plant, Professor Ito. Okay, medical biology, we have 7. And these two professors studying the immunology, okay, immunology. And these two, Kurisaki and Isotani, studying the stem cell biology. They make the normal cell to to change the stem cell, and using this stem cell, he or she tried to make a human-made organ like stomach or heart or lung. Okay, it's a futuristic study. Okay, in medical biology. He's a developmental biology, basic study. And he is cancer biology, studying the membrane protein, the controlling the structure of cell or cell shape. Okay, so sometimes cell makes many structures, but that's controlled by membrane proteins. He's an RNA biologist. His study also related to cancer. And yeah, so I picked up the Professor Kawai's. He found a receptor for bacteria and virus to initiate innate immune response. Okay, so human, we have so many receptors. It looks like the same, very similar, but many genes are involved. Okay, he's studying the total system, how innate immune system is controlled. Yeah? Very popular immuno scientist. And Professor Kurisaki, I just told you, use that stem cell, ES cell or iPS cell, use chemicals or growth hormones or many kind of conditions. He can make parts of the stomach, various different cells, and make a mini stomach. Okay, in his laboratories. Such kind of studies, quite interesting. And last one, systems biology, the microbiology laboratories, he, but actually two more scientists. So we have four microbiology laboratories and structural biologist, Professor Tsukazaki, and neuroscience, neuron cells development or neuron cells functions. Professor Besho will give a lecture after me. And he's a biotechnologist, bioengineering. I will show you that his study. And he's a bioinformatics. The use mathematics to analyze a big, yeah, the quite interesting study he is doing, many different things. So one of the microbiology laboratories, Professor Yoshida found that the bacteria which degrades or eat PET plastic, okay? And this is the first case, okay? Maybe the only case. The bacteria degrades PET plastic, and he found two enzymes which degrades the PET polymer to this monomer. So using these enzymes, we can recycle or the PET from waste to new materials or something, or we can let's say decompose it the waste of PET. They quite interesting studies. And maybe important. And bioengineering led by Professor Kato. He's an expert in the gene expression in plant cells. Gene expression means that which genes should be switched on or should be switched off. The such kind of mechanisms he is studying, and he applies this kind of knowledge to produce many important things in plant cells. For example, gamma immunoglobulin or immunoprotein are expressed in the tobacco plants or lettuce plants, or even rice. Okay, so it's very easy to obtain the immunoprotein. It's very important for a vaccine or such kind of protective methods for virus or something. So quite interesting studies he's doing. So I showed 6 cases of research, but we are, let's say, doing the very nice international education program for Japanese students and the international students. This is for mostly a PhD student, but the master students can take. We have an international bio seminar, 2-day series of 3 lectures and 1 research seminar by professors from University of California, Davis, our partner university in United States. The UC Davis, University of California, Davis, is very famous for the education in biology and biochemistry and also agriculture. And their professors are very, very good in teaching. So their class is fantastic. Okay, so the 3 lectures, students learn the background of the research the professor is doing, or history, or basic methodology. From 3 lectures, then students attend research seminar at last, and the students can practice how to make questions or how to do discussions, okay, with the UC Davis professors. And we have summer camp, get together all the PhD students, and first year doctor course students will present poster, and second year doctor course students present their research progress, oral presentation, and make up the discussions. So this is very good for doctor students. We can, let's say, share the how and what the students achieve the progress, and we can discuss each other very easily. Okay, so we can do. We have been doing this for many years, and it's very effective. So after the summer camp, D1 poster, D1 presentation in English, then they practice that other countries' PhD students. We invite 10 very best students from United States, UC Davis biology, and also very best students from China. In China, postgraduate students is in the Chinese Academy of Science. We have a collaboration with the Institute of Genetics and Development Biology in Beijing. They send 10 students. So our D2 students stay together with American and the Chinese students at the little bit isolated areas in the mountains. They for 1 week and present their research and discuss. And also they are doing some small project together in a group. Okay, so this kind of international training that our students take. And also we have used the online system. These days we can use the Zoom, but before 4 years ago, there are no such good systems. So we bought very expensive teleconference system to connect the UC Davis students to have a journal club by students. Yeah, so we are doing this kind of online journal club for students. And you can see many students, as I told you, the % of the PhD students are international students. And let's see, we very care about the Indonesian students. So these are first 10 Indonesian students studied at NAIST long time ago. You may see who is him, right? So Mrs. Ida, he runs the business in Japan. Let's say the helped NAIST to invite the good students from Indonesia one by one. Okay, so first 10 years we received, well, maybe longer than 10, but we received the very best students from Indonesia. Okay, like you and many others. But since then, the number of Indonesian students these are another photos in Japan. You can see Herman-san and Ari-san. This is a 2020 Indonesia student 8. Okay, and Malaysian students are the most popular. But let's see, last year we have 12, and number of Indonesian increasing. Yeah, and from UI we have 2 students. We have more students from UGM, but we really like to have UI students. So please think about the NAIST to be the next place for you. So we have a special recommendation program to get a chance for partner university students to study at NAIST. Okay, so we first construct a good relationship or good trust friendship with our partner universities faculty members. Then we asked to recommend good students to NAIST. So the screening process based on the recommendation, okay, by Professor Ari, and in case of the UI, and we used to be invited to such applicants recommended by our partner universities to NAIST, and ask them to stay at Japan at NAIST for 2 weeks. But these days we can do this online. Okay, after a pandemic. Okay, but anyway, we can let's say screen by interview, basically an interview. Okay, and then let's say this year pre-screening internship will be in December, and we just send let's say information about the guidance for the pre-screening internship to Professor Ari, and that will be in September next month. And we also send the application form, and the application deadline will be November, right? And let's see, the next month, September 2 or 9, 2nd or 9th, we have guidance on the pre-screening internship. In this guidance, some or 4 or 5 professors at NAIST will give his lectures, and the applicants can discuss with the international students already studying at NAIST, okay, online. Okay, so please think about the NAIST, okay, for your postgraduate study. Okay, thank you very much.
Okay, thank you very much, Professor Maki, for your comprehensive introduction to NAIST and also its postgraduate programs. And we can see several professors and also the topics that you may have interested in, and maybe you have I don't know if you want to study further in NAIST. And also, it is a valuable insight into the academics and also the research opportunities that were explained by Professor Maki. And next, moving on, we're going to have another research seminar. But before that, please let me extend a warm welcome to our honorable guests from University of Indonesia, Faculty of Pharmacy. I'm sorry that I forgot to welcome you in the beginning. So we have the Dean of Faculty of Pharmacy, Universitas Indonesia, Prof. Ari Anwar, who is also an alumni from NAIST. Please give a round of applause. And also the Vice Dean for Academic Research and Student Affairs, Dr. Apoteker Fadlina Chani Saputri. And last but not least, the head of postgraduate of Faculty of Pharmacy, Universitas Indonesia, and also an alumni from NAIST, Dr. Herman Suryadi. So thank you for attending. Okay, moving on. For the first research seminar by Professor Yasumasa Beso. So while we are waiting for Professor Beso to prepare, please let me introduce a little bit of the biography of Professor Beso. So Professor Beso is a distinguished professor in the Division of Biological Science at NAIST. And he has an extensive academic career, having served as an associate professor at Kyoto University and also as researcher at Washington University in the US. Professor Beso also holds an MD, so he's a physician from Wakayama Medical University and also hold a PhD from Kyoto University. In 2005, he was honored with the Award of Young Scientist by Max Japan. So today, Professor Beso will present his research on valproic acid induces axial skeleton anomaly via perturbations of the somite segmentation clock. Ladies and gentlemen, please welcome Professor Yasumasa Beso.
Thank you very much for coming. So this is Besho. Nice to meet you. Okay. Today I talk about the work in my laboratory, and which is in my laboratory. So I use a mouse and also the zebrafish as a model animal because we can do the genetics in these model animals. And so we have many topics. However, I will talk about the segmentation in the mouse today. Okay, so our body plan is very beautiful, and also we have a pattern, repeated pattern from anterior to posterior, like that. Okay, and so this repeated structure is conserved among the vertebrates, like fish, and also the chick and mouse, and so that means the animal body is beautifully formed from very simple structure to the very complicated structure. So during that process, they have some pattern formation. And of course, they have some genes which are very critical for the developmental process. And if they have some mutation in the critical genes, the body is the formation is disorganized. Okay, for example, if a baby has a mutation in the delta 3 or lunatic fringe or HES7, those are included the not signaling to that the mutant forms the very disorganized backbone. Okay. So, you mean, I mean, the not signaling is kind of the intercellular signaling. And delta is here. And lunatic H7 lunatic hinge are here. So that means the not signaling is essential for the axial skeleton formation. It is conserved between the mouse and human beings. In a mouse, if we destroy H7, so the backbone is destroyed like that. And also the same thing in the lunatic fringe. And this repeated structure along anterior posterior axis has some pre-pattern in the embryo. So that is called somites. Somites are arranged on the both sides of the midline, symmetrically like that. And also the somites are very same size. Okay. And so a pair of somites differentiate to the one backbone. So that means somites are the pre-patterns of the axial skeleton. And this is a mouse embryo, and this is the 8.5 days, and this is 9.5 days. So during 1 day, the embryo grows from here to so like this. So this part is very new part. And so the yellow somites are new somites. Somites formed from anterior to posterior one by one. Yeah. So I should not touch the panel, probably. I'm sorry. Okay, somites form one by one from anterior to posterior in a 2-hour cycle. So, yeah, okay. Okay. I learned it. Okay. Thank you very much. I mean, this is the new US somite is here, and the next somite is here. So it forms after 2 hours. So every 2 hours, so one somite is formed. Okay, so that means the somites are formed by periodical segmentation. Okay, in a 2-hour cycle. And so the question was, who count, what mechanism count the 2 hours? Okay, the answer was the gene expression. So gene expression changed in a 2-hour cycle like that, from posterior to anterior. And then one somites are formed, one pair of somites is formed. And after that, it's fine. Okay, no problem. Okay. And yes, the embryo repeat this pattern like that in a 2-hour cycle, and then they form somites one by one. So this is new form. So one side after one cycle, as a pair of somites is formed, and then repeat and repeat. And this is the live imaging like that. So bright color means the location of messenger RNA, like that. Okay, and I I made a knockout mice of HES7, one of the, actually I discovered the HES7 gene. HES7 is a transcription factor that inhibit the gene expression. And I make a knockout mice like that. And then, fortunately, HES7 was essential for the periodical segmentation because the resultant somites are very disorganized in the HES7 knockout mice. And the bone is same thing, so very disorganized backbone in the HES7 knockout. Okay, also, the HES7 knockout is missing the gene expression, periodical gene expression. So that means HES7 probably HES7 generate gene oscillation, which work as a clock to form the periodical segmentation. Okay, so this is a molecular model, and HES7, not signaling activate HES7 expression. Okay, and HES7, after the accumulation of HES7, increase the HES7 protein. HES7 protein inhibit its own expression. Okay, then the HES7 protein will go down because the expression is stopped. Okay, but HES7 protein is very easy to destroy. Okay, so the half-life time is 20 minutes. And after 20 minutes, so HES7 has gone, and then not signaling activate HES7 again. And then HES7 protein is accumulated and then inhibit and go down like that. So that makes the oscillation. And the period time is 2 hours. So that is the, a kind of the molecular clock to form the somite in a 2-hour cycle. Okay, so we understand that the genetic mutation destroys the morphology of the embryo. So like that. However, so you know, in this case, everything is broken. Okay, but in the other case, so this is the some we can see some anomaly in just a part of the somewhere. Okay, so in this case, almost okay, but they have some mistake like that. So we don't know why they have some mistake. In the just a not the all, just a one part. So like that. So they have it has some mistake like that. Probably, in my idea, probably environmental perturbation or maybe change of the temperature or some drug or some stress can disturbs the formation of the some pattern. But in the usual case, in the healthy body, a well-organized body shape is formed in spite of the perturbation because they have some robustness. So they are strong against the perturbation from the environmental change. But some embryo is very sensitive to the environmental change. Okay, actually, so we made another knockout mice of the NRAP. NRAP is a kind of the feedback inhibitor of notch signaling. So you know, notch signaling is very important for the somite formation. And NRAP is a member of the notch signaling, and which is the feedback inhibitor of notch signaling. Okay, and I destroy the NRAP by the homolars recombination. And NRAP knockout mice has some mistake in the vertebral bone. Okay, so this is wild type, and red color is a mistake or anomaly, and almost good, but they have a small anomaly in the body, in the backbone. And also I use valproic acid as a model of the environmental perturbation. So you know, it is very difficult to sense the environmental change, but I add valproic acid, and then valproic acid is a very good medicine for the anticonvulsant or the mood stabilizing drug. However, it has teratogenesis. It can induce the anomaly in the baby. Okay, so pregnant mother must not take this drug. Okay, and I injected the valproic acid to the pregnant mice, and the resultant baby has some anomaly in the specific point around here. Okay, and also the I injected the valproic acid to the pregnant mice, and after 24 hours, I checked the shape of the somites, and somewhere is broken. And you know, after 24 hours, the embryo elongates like that, and you know, one somite form in a 2 hour. So 24 hour, during 24 hours, 12 somites are formed. Okay. And the affected point is around 10 or 8 somites. So that means in the 0 hour, I mean, when I injected the valproic acid affect to this side, I mean, the primordium of the somites, where the gene is oscillating. So probably the valproic acid perturb, I mean, I disorganize the oscillatory gene expression. I mean, this one. So valproic acid perturb this one. And we check that one. So after 2 hours, the oscillation is disorganized like that. So that means that valproic acid transiently perturbs the synchronous gene oscillation in the PSM, presomitic mesoderm. That is the primordium of the somite. And and then I inject the valproic acid to the also the wild type embryo and the NRAP embryo, okay, NRAP knockout embryo. And then NRAP knockout embryo has more disorganized somites. So that means the NRAP knockout mice is sensitive to the valproic acid perturbation. So the bone is same thing. NRAP knockout mice has more anomaly after the injection of the valproic acid. So NRAP knockout mice is very sensitive to valproic acid. So valproic acid is a model of the environmental fluctuation. So that means NRAP knockout mouse is sensitive to the environmental fluctuation. So that means NRAP is essential for the robust system. So because NRAP knockout mice needs more time to recover. So that means NRAP knockout mice is sensitive to the environmental perturbation. So NLRP is essential for the rapid synchronization of the gene expression in the presomitic mesoderm. Okay, so a robust system of somites and axial skeleton formation includes the NLRP. So that is conclusion. Okay, so this is my laboratory. Thank you very much.
Thank you, Professor Bessel, for your fascinating presentation, a very advanced and deep research. And I think your insights are very valuable for us and also all the students here with us today. And before we go to the next speaker, I would like to inform you that we're going to have Q&A session after all the speakers finish their presentation. So you can take notes or for your questions, and then you can ask later on. And for our second research seminar will be presented by Professor Naoki Aratani. He is currently a professor in the divisions of material science at NAIST. And Professor Aratani has been a key part of NAIST since 2012, starting as an associate professor and now serving as a full professor. And his research journey has also taken him to JST and also UCLA, the United States, and he brings deep expertise in synthetic chemistry, photochemistry, and also material science with a Doctor of Science degree from Kyoto University. So today, Professor Aratani will discuss material science at NAIST from synthetic nanocarbons to degradable polymers. Please welcome Professor Naoki Aratani.
Thank you for kind introduction. Selamat siang. Yeah, very nice to meet you. Hello, everyone. My name is Naoki Aratani. Both two professors are coming from bioscience, and I'm from Division of Materials Science. So I'd like to introduce the situation of material science briefly at the first part of my talk. Okay, so this slide listed the current 20 students from Indonesia. Could you find the name of your friend in this slide? So many UI students are studying at this moment in NAIST. Of course, from Gajah Mada and also other universities. We have 20 students at this moment. Okay, and please look at some pictures. So I believe that they are so happy and enjoy the school life in NAIST. So in the Division of Materials Science, we have 7 international faculty members. So you don't need to, how to say, we can take care of all of foreigners because of their kind help. And then we have totally 23 laboratories in the Division of Materials Science. And unfortunately, I have no time to explain about all of laboratories, but I have good news. So next month, you have a chance to know about all of laboratory contents from the professors on the 6th September. So you can get the information about all of laboratories, and then at the end of this study abroad fair, you can ask the questions to directly to Indonesian student in NAIST. So we open the breakout room in this online event. Then you can enter the breakout room to ask the question directly to the Indonesian student. Starting from this event, we started pre-screening program. So in our case, after the study abroad fair, you start to prepare the application. Then after that, we are very sorry, but we only accept we can invite a very limited number of students to come to NAIST. And then the other, we prepare the online internship program. So if you have any interest in our university, please join this pre-screening program. Okay, so this slide summarizes the targets of our laboratories from the. So we do creation of academic science, new materials and new technologies by using physics, chemistry, device bio, and informatics. Especially, recently we built a new center, data science center, and the data science center offer the NAIST education program of data science. If you choose the data science education program, you can learn the data-driven science or artificial intelligence and so on. So if you have any interest in data science center and the data science education. So please ask, please join the press screening program. The professors join this abroad fair. So you can ask the question directly to the professors. Okay, and then this April, we built another new innovative research center regarding the medicine and medical aspect. The name of center is Medilux Research Center. So Medilux is artificial word. So the meaning is medical with optical things, so such as imaging or sensor. So we collaborate with many medical schools nearby, NAIST, Nara Medical University, Kinki University, and so on. Then we will do the healthcare project, including imaging, AI, soft material, and sensor, and so on. Okay, this slide summarizes the recent topics from chemistry labs. This is my lab, so I will explain about this later. And then Professor Kawai is making the photoresponsive materials such as photochromic compound, which can change the color by light from colorless to colored such as blue or yellow. Then it's reversible. So after the changing the color, if we add another light or heating, the colored compound go back to colorless compound. This is called the photochromic compound. And Professor Lappen is the close appointed professor. He spent half a year in Japan, half a year in France. He is making nano size car. Nano means the molecular size car, and he drives this small car on the flat substrate. And actually, he is a organizer of World Championship of Nano Car Race. So he organized the Nano Car Race twice. And then this is research topics of Professor Ajiro. He is a polymer chemist. I will explain later. Okay. So our research is supported by Center for Material Research Platform. It's in the same building. So we have 55 instruments, and it's covered from the physics to chemistry and biology. So and all of instruments are operated by technicians. So you can submit your sample to technician, then technician can analyze all of properties, then go back to your sample. Or if you have interest, you can have the lecture from technician, and after the lecture, you can use these kind of instruments by yourself freely. Okay, so let's move to the research topics. So these are major three materials: ceramics, metal, and polymer. And among these, only the organic polymer is soft and light. So we can use these kind the polymer polymeric materials in our life. So it's indispensable in our lives. Professor Ajido is making the conductive polymer, thermal responsible polymer, and then pH responsive polymer and a biodegradable polymer. He is Professor Ajiro, and actually, he is my high school mate by chance. This is young Ajiro, and this is young Aratani. And then he is making biodegradable polymer, highly biocompatible functional polymers do not generate any acidic compound in and after hydrolysis. So his material can be used in our body. And for example, if we make the stent by his material, it's we can extend the blood tube without toxic material. Also, biocompatible functional polymer is high compatibility at ambient temperature. He can control the properties of polymer by choosing the monomer. So he can design the final properties of polymer by choosing the monomer. And so I promoted full professor on April of this year. So I just started a new team from this spring. So I just have only a few students. So in our life, we have many carbon materials such as diamond activated carbon and graphite. And also in the research field, we see the fluorines, carbon nanotubes, nanorings, and so on. And most familiar carbon material is graphite. So you know the material of pencil. So one atomic layer of graphite is graphene. This is, I think it's the most superior material in the world because the mechanical strength of this one atomic carbon nanosheet is 200 times stronger than steel. It's the strongest material, even made from carbon. So it's soft and strong. And this material has ultra high mobility, more than steel. However, it's very difficult to control the properties because the size and the terminal structures are not uniform. So if you peel out one layer of graphene sheet from graphite, so each layer has different property. So that's not, we cannot use such a material in the electronic circuits, so on. So my research is to create the discrete nanocarbon materials from the small piece of polyaromatic hydrocarbons, such as naphthalene, anthracene, pyrene, or pyrene. So from the by using the bottom-up strategy, we can make the discrete skeleton of nanoparticle. This is a crystal structure of carbon nanosheet composed by 26 sp2 carbon atoms. This one molecule can release up to 5 electrons. So it's possibly used for the battery. So, and it's completely reversible and enough tolerant. And next target is infrared, near infrared dye. The characteristics of near infrared light is accounted for the for the 40% of sunlight. So if we can manage the near infrared light, we can get the energy from sunlight more. Now, for example, the organic solar cell throughout the 40% of near infrared light. So if we can make very efficient near infrared dye, we can get energy from the sunlight more. And also, our body has blood, it's colored, so red color, so hemoglobin can absorb the red light. So if we can manage the near infrared light, it's highly transparency and high safety for living organisms. So if we can build the medicine based on the near infrared dye, we can we can excite that dye inside our body without any interfere of blood. Okay, so this is a fluorone, which is a highly colored compound. It's the color of this one is pink. So this solution color is pink, and it emits pink light. I added 2 more benzene rings to this fluorone, and then the result is as shown here. So this compound emits from 800 nanometers to more than 1,000 nanometers. And our eyes can sense from 400 nanometers to 700 nanometers. So this is completely invisible. So, so this compound emits colorless light. So we can use this material for sensing. And then so next topic. So I'm making the carbon nano ribbon. So for 30 years ago, German chemists make the nano ribbon up to 1, 2, 3, 4, 5 naphthalene unit. This is colored. This is this can absorb the light from ultraviolet to 750 nanometers. And so this is a green dye. And then I added 1 more naphthalene unit to this compound 2 years ago. This is a crystal structure of 6 naphthalene fused compound. The color of this compound, you see almost no color, because this compound can absorb the 95% of near infrared region. But if we make the high concentration of solution of this compound, it's like brown color. So quite recently, I added 2 more naphthalene unit to previous compound. Then it's now the absorption of this compound located in the near infrared range up to 94%. So it's almost completely colorless compound, but even colorless compound, this can absorb the solar light without any interfere of our vision. Okay, so we are making many kinds of nanocarbon materials, from carbon nanosheets to insertion of a heteroatom or eight-member ring, or not only the planar compound, we are making the tubular compound or layered compound to show the very characteristic properties of nanocarbon atoms. So thank you very much for your kind attention.
Okay, thank you for Professor Aratani for the enlightening presentation, and also the depth of your research is truly impressive, and we surely grateful that for the knowledge that you shared today. So for the next sessions will be the Q&A sessions. You can ask from our three speakers here. Is there any questions from, especially for the students? Well, I have several questions actually, but I think this is a good opportunity for the students first, if you want to ask. Or maybe you can ask in Bahasa and I can translate it for you. No one? Okay. While waiting, maybe you're still shy. While waiting for questions from the students, actually I have a questions for Professor Maki. Okay. Is there any special reason why NAIST is only established as a graduate university, postgraduate university, not like you don't have the undergraduate program, right? Is there any reason for that?
Yes. That was almost 40 years ago. Japan economy was the top in the world, and the United States always complained Japan utilized the knowledge or some new technology invented by Western countries to get more money. So United States always ask Japan to create good education system to, let's say, foster the good scientist or engineer. Then our government decided to change the national university to strengthen the postgraduate study. But you know, the big universities cannot be changed easily. So basically, that big university, like University of Tokyo or Kyoto universities, are for undergraduate educations. So faculty and divisions or such kind of structures are for the general structure of our knowledge of science and technologies, always divided and not changed. Okay, that covers all the areas. But for the research, especially postgraduate educations, we think of the research, not the general knowledge of science. So our government decided to construct very new independent graduate school in Japan. The first one was that Hokuriku Science and Technology Institute of Hokuriku, called JAIST, okay, Japan, JAIST, in the north part from Nara. And after 1 year, NAIST was settled. And let's say these two institutes focusing on the cutting edge area for industries or societies like IT technology or material science. And biological science is only at NAIST. That JAIST, Hokuriku, has the knowledge science or something quite new. things, but let's say from such reasons, our government invested a huge money to build our institute. So we, let's say, were very lucky to set up all the equipment or facilities. Yeah, we can use a huge amount of money. Okay, but let's say we have to get students from all over the Japan, and maybe in 15 years, we were good in getting the good students from all universities in Japan, but government or decided to, let's say, change the big universities to strengthen the postgraduate study. So that Tokyo or Kyoto universities or other major universities becomes the graduate school, not the undergraduate university. Okay, so then we compete with the many major universities in Japan, but we are doing well. We are attractive to some the active students, and also 15 years ago we started to get more international students to let's say make our education environment more international because we use English for the education. That was 15 years ago. So when you were there, everything is Japanese. So international students must learn Japanese language, and the screening process requires that there's some capability speaking or writing Japanese. That was a little very yeah, tedious and difficult things. But 15 years ago, we decided to change. Our university is more international. So one thing is we made a good collaboration with the UC Davis, California University or Davis. And we have collaborative education programs developed. Actually, our PhD students stayed UC Davis for a month to, let's say, get training in conversation or small project at the laboratories. Then, let's say, we started to recruit the international students from Asian countries. We're very lucky. Indonesian students already graduated, and they becomes the very important persons in the Indonesia, and they have been helping us to get very best students from Indonesia. That's the reason why we are just a graduate school. Okay.
Okay, very thank you for a very thorough answer. So any questions from the students? No? Okay. No? Well, I have another question. Can
I may give some information for students who may be interested in studying at NAIST. So our pre-screening internship used to be, let's say, face-to-face interview. We invited the applicants to NAIST for 2 weeks, and during the stay, the applicants must give the presentation, maybe 15 or 20 minutes for their thesis work or what they are interested in or what they learn. And the 3 professors will evaluate the willingness or capabilities or let's say, personalities or something like that. Then the applicants visit three different laboratories which the applicants are interested in. Then the meet professors or faculty members or students at individual laboratories, and with the professors, the applicants will have an interview, and then professor and applicants evaluate each other. So applicants should choose which laboratory is suitable for their postgraduate study. And also the professors will evaluate the suitability of applicants for the postgraduate educations. So after the pre-screening, we will inform the result. If we can make a good matching between the applicants and the professors, then we, let's say, make a list depend on the scores of the applicants' performance. Then let's say we decide who should be incorporated in the NAIST. And let's say next important thing is that financial support. We have 10 slots of financial supports, the mixed scholarship and the NAIST scholarship, plus some other extra the financial supports, maybe 2 or 3. So we always ask that successful applicants to apply embassy-based mixed scholarship. These days, 2 or 3 successful applicants get the mixed scholarship through embassy, a very high successful rate. So totally 15 to 17 applicants will come to NAIST. Okay, so that screening process. So yeah, that's probably enjoyable and try. Okay.
Yes, I hope I know students here can try.
Okay, thank you for. So I will add your information because NICE give me trusting for as a representative. So for recommendation ya. So I will talk in bahasa ya. So saya diberi kepercayaan oleh NICE untuk sebagai perwakilan gitu ya, untuk memberikan rekomendasi yang mau ke NICE. Jadi tadi dikatakan the applicant should not send the application directly. Gitu. Yeah. So to me, yeah. So I will select it first selection under my decision. So silakan mengirimkan aplikasi, kemudian saya akan menseleksinya sesuai dengan nanti apa namanya profesornya dan lain-lain. Kemudian dari semua aplikan yang masuk, saya akan kirim beberapa yang terbaik untuk dikirimkan ke sana. So I will send maybe 4 or 5 best applicant from maybe various, maybe 20 applicant come to me. So I will select, then send to the best to the NICE. So based on experience, so almost every years I send. Then yeah, last year Ayun, Ibu Ayun, Pak Rezi, Pak Catur sama Pak Indra, itu Pak Indra dan Pak Catur sudah kembali, itu saya merekomendasi beberapa dari fakultas, dari MIPA juga ada, from Faculty of Science, based on my recommendation also. He back from the organic chemistry from Kakyuchi Sensei. So yeah, so thank you for opportunity to me to have a representative from material science and also bioscience. So bioscience also last few week send me many application form to be filled. So if many student interested, so contact me and I will send you the application form, then fill and return to me. So I will pass through the NAIST under my objective selection. So yeah, thank you very much. Yeah.
So, because of the limitations of our time, sadly, we have to end our Q&A session. But you can always ask our speakers outside this event, right, Professor? Yeah. Okay. Next, to commemorate this special event, we will now proceed to the exchange of placards and souvenirs between the Faculty of Pharmacy, Universitas Indonesia, and also NAIST. So we kindly invite Prof. Ari and Prof. Maki. Okay. Okay. And also Professor Besso and Professor Ratani, please. Oh. Okay, next we would like to invite all our speakers and also our honorable guest for a group photo. Okay, and with all the students, please come forward. Okay, that ends our event today. We hope you find today's event very informative and oh. So, NAIST have something for you, the students. Yeah, wow. Yay. Yeah. Okay, we will give all of this to the students. Maybe after we conclude the event. Is it okay? Okay. So, okay. So everyone, that concludes our international guest lecture series for today, and we would like to extend our heartfelt thank you for our speakers, Professor Maki, Professor Aratani, and also Professor Beso. And thank you for coming for the students. And we hope that you found today's event both informative and also inspiring. Thank you. And we look forward to seeing you at the future events. Have a great day. Thank you, everyone. You can come and yes.